My project today is I made a theoretical CRISPR design for the disease PKU. So a bit of background on what CRISPR is. Um, it's gene editing that can selectively modify the DNA of living organisms. So CRISPR stands for clustered regularly interspaced short palindromic repeats, and Cas9 is the CRISPR associated protein that does the editing in the genes. And this editing was actually adapted from a bacteria's immune defense system. And essentially how it works is that a small RNA piece has a guide sequence that will bind to a specific target sequence in the cell's DNA. It'll also bind to the Cas9 protein. Once inside the cells, the sgRNA, which is the guide RNA, will recognize the target sequence, and the Cas9 protein will cut the DNA at that location. And once cut, the cell's own DNA repair mechanisms can be used to add or delete genetic material, or a custom sequence can be added in as well. This system of editing won the Nobel Prize in 2020 as well. So a bit of background on the phenylketonuria, and it's an inherited disorder, which is autosomal recessive. That means basically that autosomal means this disorder is in a body chromosome so it's not sex linked, and recessive means that you need two recessive alleles or mutant alleles in order for this disease to take place. So what happens in PKU is that the body is unable to break down the amino acid phenylalanine in your liver, and so the levels of this amino acid will continue to build up in your blood and brain, which can cause brain damage. And other symptoms such as epilepsy, an unpleasant odor, eczema, and then frequent sickness. For the current treatments, most people have managed to live a good life by eating a low phenylalanine diet, but there isn't a permanent fix yet, and PKU affects about 1 in 1,500 newborns. On the figure on the right, you can see here that high protein foods contain a lot of phenylalanine, and so when children or anybody with PKU eats high levels of phenylalanine, then it's not able to be broken down, and so it builds up in their brain. And normally, it gets broken down into tyrosine, which gets broken down into hormones like dopamine, serotonin, and then melanin. So, for the specific mutation I'm focusing on today, essentially it's in the PH gene, which is the gene that provides instructions for making the enzyme phenylalanine hydroxylase, which is the enzyme that breaks down phenylalanine. And mutations in this gene are what cause PKU, because mutations end up leading to altered enzymes which can't break down phenylalanine. And so the mutation I'm focusing on, and the most common mutation as well, is R408W. R408W, basically the R stands for arginine and the W stands for tryptophan, which are both amino acids. And what happens in this mutation is that arginine is replaced with tryptophan at position 408. So what happens is normally the codon, which is a set of 3 nucleotides, it's normally supposed to be CGG, which codes for an arginine, but in the mutant codon, the C is replaced with a T and it becomes TGG, which is a tryptophan. The reason why CRISPR would be a good solution to this disease is because there's a single base pair mutation, which makes it easy to edit, and there's potential for a one-time permanent fix this way. So for the editing strategy, our goal is to convert TGG back to CGG at codon 408, and we can do this by using adenine base editing. Adenine base editing is a genome editing technology that allows the conversion of adenine to guanine in DNA. And in order to do this, the Cas9 protein needs to fuse to adenosine deaminase, which will help find the DNA target along with sgRNA. So how adenine base editing can help fix this mutation is the R408W mutation changes the C in the codon to a T, and all nucleotides are paired with they're matching base nucleotide. So C and G are usually paired, and then T and A are usually paired. And so when the mutation changes from a C to a T, that changes its paired base from a G to an A. And so when the newly fused enzyme, the Cas9 and then the adenine deaminase, when they combine and they convert the wrongful A to I, which is enzyme, that I is read as G during DNA repair. And so when yeah, and then so when like the DNA is repairing itself, it reads the I as G, and so it pairs it with its rightful nucleotide base, which is a C, therefore correcting the TGG codon to CGG. There's a so PAM is basically a DNA sequence. Um, yeah, PAM is basically a DNA sequence next to the target site that Cas9 needs in order to bind. And this and NGG basically means N. It can be any nucleotide, A, C, T, or G, and then two Gs. And since there's not really a clear PAM sequence next to the mutated A, we need to use a PAM relaxed Cas9 variant instead, which basically means that it's more flexible with what it accepts. It can accept NG rather than just then N and then two Gs. This is optimal. This type of editing based editing is optimal because it's highly targeted. It has a small editing window. There's a single stranded break. It breaks only one strand of the DNA instead of both strands, which leads to a lower chance of mutations. And it's also a single base editor, which is perfect because we have a single base mutation. On the figure on the right here, you can see that adenine will change, that adenine base editor will change adenine to inosine, which is read as a guanine during replication. So so sgRNA, again, it's a piece of RNA that guides the Cas enzyme to the spot in the DNA that will need editing. And so we need to design the sgRNA in order to guide the base editor to the PKU mutation to change A to G. So how we design this is we have to find the real PH exon 12 sequence near the mutation R40W, and an exon is basically just a section of DNA that contains the nucleotides that actually code for things. And then we need to find the template strand of the DNA, which is the DNA which is a strand of DNA that codes for that RNA will copy. And then we have to pick around a 20 base pair target that has the mutation A and the PAM next to it. And so I ran this through a website called ChopChop, which can help identify sgRNA designs. And this is an example of a sequence that can be used for sgRNA. And then we need to figure out how to deliver our sgRNA and base editor to the liver cells. And so we need a delivery vehicle in order to do that. And we can use lipid nanoparticles, or LNPs, which are tiny fat bubbles that contain mRNA that can encode the base editor and the sgRNA. And a good thing about this is that liver cells naturally take up LNPs, which makes it optimal because it targets liver cells. The mRNA degrades quickly, which means there's a less chance of unintended edits, and it's also easy to redose if necessary. Sometimes CRISPR and base editors can bind to similar DNA sequences, which can cause unintended edits, or also known as off-target effects, which can lead to unwanted mutations. And so ways to prevent this are bioinformatics tools like ChopChop, as I mentioned earlier, can predict possible off-target sites genome-wide. And it's always important to test your sgRNA in a cell culture first. So after the CRISPR design, the expected result should be conversion of the mutant TGG or tryptophan back to CGG, which is arginine, at codon 408 in the PAH gene. The restored arginine in position 408 will lead to a functional PAH enzyme, An increased pH enzyme activity in the liver cells will lead to normal phenylalanine breakdown. And because there's reduced phenylalanine levels in the blood now, it'll prevent neurotoxicity. This is my works cited. Thank you so much for listening.
Excellent, Anika.